The overall goal of this procedure is to detect RNA transcripts in place within cells and tissues.
This method can help answer key questions in the cell and developmental biology fields, such as where inside a cell, a tissue, or an embryo an RNA is located. The main advantage of this technique is that it detects RNA spatial distributions with both high levels of resolution and sensitivity, and at a low cost compared to other methods.
We initially developed this protocol to study the subcellular distributions for as many Drosophila RNAs as possible. This was in order to determine the extent and diversity of subcellular distributions.
We started the project working with the embryo, but later adapted and optimized the protocol so we could work in high throughput with tissues dissected from larvae and flies.
The most difficult aspect of the procedure is the number of steps and the consequential potential for error, as well as the need to ensure the absence of RNases during probe production.
To facilitate throughput and cost effectiveness, the procedures demonstrated in this video utilize 96-well microtiter plates for all steps. It is also easy to adapt this method to smaller sample numbers by cutting the plates into 8 or 12 well strips. Start by diluting 10 microliters of an overnight bacterial culture in 90 microliters of autoclaved double distilled water in each well of a new 96-well PCR plate. Denature the DNA at 95 degrees Celsius in a thermocycler for 5 minutes. Immediately place the plate on ice for at least 5 minutes. Set up the PCR reactions in a new 96